Hello there, this is Salim again, and tonight we are gonna try to do a small video about decontamination and exploring the sample. So I'm gonna reuse again the sample we were using in the last video, and I have it preloaded, and this is pretty much what we had before. And we had these cell types, more or less. That's what we could refine to this point. And we have these cluster IDs. There were plenty of them, which help us to identify some clusters and their gene signature. And before, I pointed out that it's possible that this sample is contaminated because everything is EpCAM positive. That means all those here are colonocytes, exactly what we are looking. But if that's so, if that's so, we shouldn't have that many fibroblasts in here. We should have only absorptive stem cells, progenitor cells, goblet cells, and everything is pointing out to that. So the thing is that what we're gonna do is click in the external, and for this point, at this point, we should have the R environment set up and run detect and bind contamination. And we hit on it, and it might take some time. Oh, sorry. Here, continue. Use the default for storing data temporarily, and that might take quite some minutes, within 10 to 20 minutes, depending on the size of the sample. It's gonna call an R function and it's gonna do decountx. It's a really good pipeline we usually do in the single cell data core. And it's computing it. I'm gonna stop it until it finishes. Okay, it finished, and it will ask if we want to remove the contamination. We hit on yes, contamination removed. It's not gonna show anything. It's gonna be weird. Here in the backend, that's what it happened. It called decountx through the package Zelda in R, and it does some iterations and cleans up the UMAP and modifies a little bit the cone matrix. What we end up having is what we saw, and what we have to do for looking at the changes is executing again the QC filter, similar quality as we did before. In this case, we did astringent before. We are gonna do astringent this time as well. And then we do that, and it's gonna take some few minutes, seconds maybe. Gonna stop it for now. Then it finished, and we get that is reduced from 17,600 genes to 17,200, and cells reduced by 600 around. We accept. And we should have something similar as before, but if we do the UMAP again, should look cleaner. Using existing, no. We are going to regenerate the UMAP 2D. Utilizing, in this case, I want to use all the genes. I'm gonna stop it and come back when it finishes. This is what we end up getting. If you see it, now it looks like more separated to every single of them. And here is something I wanted to look at before. The EpCAM expression again. All right. If we look at again, it cleaned up some regions. For example, here where macrophages was, no macrophages, no. Some epithelial, which is not good. There should be a lot of EpCAM expression in epithelial cells in the colonocytes here. So what it happened is that it didn't help. What this is pointing out is that potentially it is a really low quality set. So what we gotta do, it's just again look at the violin plots. Where is that? Violin plots of QC metrics. And after it pops up, we can see that the expression library size is very small. Mitochondrial is small, which is fine. and number of genes is 400 approximately. So it seems that is quite low quality. So I'm gonna save it just to keep a track of it. I'm gonna call it NoQC custom annotated. And here I want to show one thing before closing this and doing the process I want to do. If we do hit here in the heat map marker and then the heat map, now to the Panglau, and I want to look at the cell type, method DE, this is what I tried to do last time. It's gonna do the heat map of the gene markers. Don't order, A and B, normalization, then row wise, and we are gonna use standardize it. We can explore it with different, oh, didn't work, huh? Should I try something else? It's supposed to be fixed. Wait a second, I'm gonna see if I have the latest version. Alright, seems like the gene marker heat map still not working properly, needs to be debugged. For now, we just keep it like that, and we're gonna revise that later. I'm gonna come back to now the quality of the sample. So we had these many cell types this time, and I'll just show again that the automatic Panglau annotation brings just few cell types, and once we process again the data, we're gonna have little different things. That's what I want to show. Might finish soon. That's it. We merge, and that's what we get. It seems similar to what we had before, just a little different. Then I'm gonna close it, clear the environment, and what I'm gonna do is open again the samples that I stored at the very first with no QC or anything. I recommend you anytime to keep a track of all the files. So I'm gonna open MATLAB. I'm gonna start, instead of the QC, I'm gonna start with no QC at all, just completely raw. Union is fine, intersection is fine, it's gonna keep the same number of genes. Okie dokie, it gets 33K and 14K cells. This time we are going to look into first the quality metrics, and as we see, it's similar, 4,000, 1,000, and mitochondrial, or also around 5%. What we are gonna do this time, it's QC filter, but with relaxed. It's something we shouldn't do, and especially with the newer data sets, should come with better quality. So in this case, we just hit on it. Oh my god, it shouldn't. No, I'm gonna stop the video. All right, sorry for that. Just open R. Now we had this from 33,000 to 90,000 genes, and from 14,000 cells to 11,000. We accepted changes, and we are gonna work with this. If we do the UMAP 2D, upper right, yes, all genes, and gonna stop the video until finish. And this is what we get. It's a different shape. And I don't know if this is better or worse. We need to figure out. And the second thing I'm gonna do is I'm gonna save that into the workspace. Save the SEE, and should show in the workspace. And I'm gonna do the latent clustering, SEE, and the resolution of 2. Gonna hit on it and come back when finish. Okay, it finished, and we have the object, so we load it. And if I get the cluster ID, should have the numbers. Let's wait if it, okay, it gets it. And we can annotate with Panglau. And okay, it's human and automatically. Gonna stop it for now. Okay, it finished. Now we merge the cell types. and we have little more cells. Indeed, we got quite more B cells. I'm sorry. And now we kept some T cells. So it is important to be careful with this. And now we don't seem to have, no, we have the drift. If we look at the sample, batch ID, we see the blue separated from the orange. It is what it is. So we come back to cell types. And now we are gonna try to run again decontamination, and with that we should see a better separation within all these populations. That's part of the decontamination, and especially now that we kept lower quality cells. So we're gonna run the detect ambient RNA, continue, use that, yes, and gonna come back and finish. Alright, it finished, and then we hit ingest to accept the decontamination, and we are gonna do again QC filter, but as much as we did relaxed, we stick to relaxed. Otherwise we are gonna lose a lot of cells. What's happening with this other? I have 2 copies. I'm gonna close that, and it removed 500 cells. It's good. And now if we redo the UMAP, use existing no, UMAP to the overwrite all genes, and we let it be. Coming back in few seconds. And this is what we get. Now it seems to be less separated, and that is good. That's what I am used to look at this kind of samples. It looks a little better this time. And here we are gonna do one last thing. We are gonna redo again the latent clusters, because now the cells are decontaminated. We are gonna have different community of cells. So we are gonna do the latent cluster again with resolution 2 and see what happens. Gonna come back. It's done. Then we're gonna open again the object, and we supposedly have different communities. We're gonna look for cluster ID. It's gonna take some time, and we get these new communities. We are gonna do again the annotation with Panglau human automatically. Yeah, it's getting a better shape. And this is what we get. It looks fine. B cells, fibroblast, endothelial, epithelial here. And here for last, we are gonna try our previous marker set. You're gonna hit on cluster ID, and then you're gonna annotate with customized markers. Here, I'm gonna paste the markers we built before, and I'm gonna include the B cells and T cell markers. Gonna stop it and come back when it finishes. Oh, it's about to finish though. Yeah. Okie dokie. This is what we got. Stem cell cancer progenitor. It looks very similar to before, but now, for example, we have these 2 communities. This unknown is here. This unknown is here. I think this is fibroblast. Potentially this is fibroblast. No, this is fibroblast here as well. Here is stem and cancer stem cell. That's what we got. If we look at the batch ID, those are separated still. And cell type. Apparently in the left one, we don't have immune cells. although the T cells are gone, but still was only lighting up here. So again, we have epithelial and also all these epithelial. This annotation can be refined even further, but I might say it is the iterative process we start before. We can anytime roll back to the Panglau annotation that's very stable after we have the clusters. Gonna come back soon. Well, now we merge it, and yeah, it looks like we have here the B cells, enterocytes, fibroblast, these endothelial cells, endothelial here, fibroblast here as well. These are two different type of fibroblast, so we will have to find the markers for the second population here, and the epithelial here, and the enterocytes. What's the difference within these two? And that's it. Podocytes, that's potentially the cells we couldn't identify here, or we didn't even have neuron markers here, so that's probably the unknown population, but those are not neurons, are something else, and we have to figure out the markers by, again, brush mark the E expression and find what that could be. So far, I think we finished this set of videos, and the only left video is gonna be related to differential expression analysis downstream, where we are gonna do the GOBP analysis, and we are going to do the gene agent analysis. And that's it for today. Hope you enjoy it and have a good night.